Thank you very much for inviting me to present this afternoon. My name is Richard Godfrey, CEO of BerGenBio, a biotech company from Norway. The usual disclaimers and forward-looking statements that I draw your attention to. First of all, for those who don't know our company, maybe I can just give a brief introduction. An oncology biotech company really focused on developing inhibitors of a protein, a tyrosine kinase, that we discovered drives aggressive cancer. This protein is called AXL. It sits on the cell surface and it drives the properties that make cancer aggressive. Aggressive cancers are ones that no longer respond to drugs, that are invisible to our immune system and that spread around their body. These are the aggressive cancers that really do cause death. We've done an awful lot of work understanding the role and function of AXL, and today we really are the world leaders in understanding the AXL biology. We know that AXL has a strong correlation with immune evasion, with PD-L1 expression, and also the activity of the dendritic cells, which we'll come back to. In addition to developing therapeutics, we've also developed companion diagnostics. AXL is not driven by a mutation, it's an epigenetic upregulation, and we can identify the patients who have an upregulated AXL protein. Today we have 3 AXL inhibitors in clinical development, an orally available highly selective inhibitor of AXL called bemcentinib in phase 2, an anti-AXL antibody, and an anti-AXL ADC, which is a partner program. We have demonstrated quite compelling proof of concept with bemcentinib in AML and in combination with chemo and Keytruda in non-small cell lung cancer. As a business, we're listed on the Oslo Stock Exchange. We maintain a research group in Bergen and an administration group, but most of our operations take place from Oxford in the UK. We're 38 people in total, and we closed Q2 with 324 million kroner on our account. So I mentioned a little bit about AXL, and I'll just remind you of what it is. So here we have a cancer cell on the right-hand side there, and you can see AXL, a typical tyrosine kinase protruding on the cell surface. And it drives these aggressive traits of cancer. It's upregulated when the environment around the tumor becomes hostile. It's poisonous with chemo, it's hypoxic, it's starved of oxygen, or there's lots of immune infiltrates. And it allows the cancer cell to go through an epithelial to mesenchymal transition and become a dangerous, robust cell. But what we also understand now is AXL's present on the dendritic cells, the NK cells, the macrophages in the immune system. It's also upregulated there where it suppresses the immune response. So it's a multifaceted mechanism of survival that cancer cells use. And say because it's epigenetic, it's dynamic as well. So what we can see is cancer cells can survive chemo, they can move around the body, and they can hide from the immune system. All of that is mediated by AXL, and by blocking AXL, we can reverse that immune suppression, and we can make cancer cells sensitive to drugs again. And there lies the foundation of our clinical hypothesis. So bemcentinib, there we are, very straightforward small molecule inhibitor of AXL. The magic here is it's highly selective. It's not a multi-kinase or a pan-kinase inhibitor. It's highly selective at AXL. AXL is only upregulated by aggressive cancers, and therefore the on-target toxicity is very low, and the off-target toxicity is, well, there is none. Today we've manufactured it at multi-kilo scale. We formulate it as a simple capsule, and it seems to be very, very robust as a drug product. In terms of its safety, we've now administered it to more than 250 patients, and it seems very well tolerated as a monotherapy, but also in combination of all other drugs we've tested it with. Our phase 2 program is quite extensive. Tested as a monotherapy, combination with chemo, combination with targeted, combination with IO agents. And you can see a whole list here, some of which are ongoing, some of which are now complete. And at this time in our development cycle, we're seeing very, very clear opportunities in acute myeloid leukemia, particularly in a second-line setting, and in non-small cell lung cancer in combination with immune checkpoint inhibitors. We have an extensive collaboration with Merck, combining bemcentinib with Keytruda in breast cancer, lung cancer, mesothelioma, and a few other indications that are yet to be announced. So it certainly has a lot of promise. Acute myeloid leukemia. I think we've known of this disease for a long time. It's still an orphan indication. In spite of recent innovations, it's still a terribly unmet medical need. We have reported proof of concept clinical data as a monotherapy, and also in combination with low-dose Ara-C, which is a low-dose chemo agent which is used quite extensively to treat these patients. In spite of it being an orphan indication, it's still quite significant in terms of size. And although in spite of recent innovations, particularly drugs such as venetoclax, we still see that the 12-month overall survival is very poor, but the 5-year overall survival rate for these patients is very, very poor indeed. Indeed, the indication is positioned for significant growth in coming years, and primarily that's driven by innovations such as ours. Our proof of concept data is quite similar, both as a monotherapy and in combination with chemo, with one exception. And as a monotherapy, we must identify the patients that are AXL positive, which we can do with our biomarkers and companion diagnostics. Whereas in combination with chemo, it seems to be equally effective across all patients. At this time in our development, we're quite satisfied with our proof of concept in monotherapy, but we think we need to do some more validation of the data in LDAC combination, and that's the stage that we're now entering into now. In non-small cell lung cancer, in particular in combination with Keytruda, we've shown quite impressive response rate in the second-line setting. Overall, a 27% response rate, vis-a-vis Keytruda maybe 10 or 12%. In AXL-positive patients, this time determined by our immunohistochemistry assays, we see a 40% response rate with a median overall survival of 12 months. Again, compared to Keytruda, which maybe shows 10-12% response rate and really no overall survival benefit whatsoever. I think everyone in the room realizes that non-small cell lung cancer is a very large and significant disease, and in spite of recent innovations, including that of Keytruda, still remains a very unserved patient population. Indeed, what we may have seen even at ASCO just a few months ago, some headline data that patients taking non-small cell lung cancer patients taking Keytruda see something like an 18% 5-year survival rate. The small print here is that's only patients with the high PD-L1 score. In other words, the patients with the biomarker for checkpoint inhibitors. When you look more closely at that data, what you can see is the patients with the low and 0 PD-L1 score, which represents 70% of the patients, are seeing a 5-year survival rate of just 12% or 3.5%. Really no improvement over what they would have seen with chemo alone. So our studies this far have been in second line. We've completed the second line study in patients that are chemo relapse, patients that have failed chemo and now take bemcentinib plus Keytruda. And we reported out some of that data at ASCO, and we'll continue to report out the balance of that data towards the end of the year. Here, quite convincingly, we see that the vast majority of our patients are in this low or negative PD-L1 patient population, and 56% of our patients, in actual fact, were AXL positive. You can see very clearly there that we're looking at half of 70%, 35% of patients that are PD-L1 low or negative, and AXL positive. That represents a very large patient population. But we're also exploring in second line the patients that have already taken the checkpoint inhibitor and failed, the so-called salvage study. Now, we realize this is a very tall order to ask of a drug to reverse acquired resistance to an agent. If we're successful, we see this second line position as a very big opportunity and potentially a more rapid route for us to progress. But even as we stand here today, pending results of that salvage study, what we can see is that bemcentinib and Keytruda presents a very credible challenge to Keytruda and chemotherapy. And bearing in mind that our study here is in second line vis-a-vis the performance of Keytruda chemo in the first line. So watch this space as we unveil our clinical plans going forward. Essentially, our objectives now in the next 9 months are to confirm whether we pursue AML as a monotherapy or in combination with LDAC, and to confirm whether we go first line or second line in non-small cell lung cancer in combination with Keytruda. Just a few words on our other assets. Anti-AXL antibody is a wholly owned asset that we developed ourselves. It's coming to the end of its phase 1 healthy volunteer study right now, and we'll announce the design of the phase 2 study towards the in quarter 2 next year. At this point, we can confirm there's no significant safety observations in the healthy volunteer study, which is exactly what we'd expect. And our partnered ADC program with a Swiss biotech company known as ADC Technologies is also in phase 1. The only thing that I can report here, as it's an agent that's not under our sponsorship, is that it's entered phase 1 and there's been no red flags reported. A few words about our finances. Typical of probably most biotech companies, the vast majority of our expenditure is in R&D, and our expenditure seems to be fairly constant over the last 6 quarters. Similarly, our cash position is reasonably constant, hovering around 300 or 400 million kroner on our account. I will draw your attention to analyst coverage. We have quite broad analyst coverage. The two at the top, Wainwright and James Trading, are US banks who have picked up our investment case and covered that. I think you probably know ABG, DNB, Arctic Securities, and Trinity Delta is paid for research that we sponsor out of London. News points to the rest of the year at the usual cancer congresses. So we'll be presenting some more data at World Lung in a few weeks' time, at ESMO at the end of September, at the Immuno-Oncology Symposium, SITC, towards the end of November, and the American Society of Hematology at the end of the year. So that, of course, will focus on our lung cancer data and our leukemia data. And that's it from me. Thank you very much.